Hello everybody, welcome to our webinar this afternoon. So before we get going, just some small housekeeping bits. If you have any technical issues during today's webinar, please just send a short message in the question box on the right hand side of your screen. And during the webinar, we would like to invite you to ask any questions. And to do that, you can use the question box, which is also on your right hand side. And then at the end of the webinar, time permitting of course, we'll try to answer as many of these as possible. And any that we don't get around to today, we will answer offline and share them with you along with a link to the recording. So today's webinar is called On the Prowl, What to Look For During a Contamination Control Audit of a Pharmaceutical Plant. So my name is Rebecca McManus and I'm Account Sales Executive at RSSL with a primary focus on sterile and non-sterile microbiology. I've been a part of the company for just 2 years now, and I moved to our commercial team at the beginning of this year. So my role is to support clients that require a variety of microbiological testing, from routine inquiries, so like microbial limit tests, to more complex things like environmental monitoring or cleaning validation projects. So for those of you who are new or maybe haven't heard of RSSL before, who are we? So RSSL, or Reading Scientific Services, are a global CRO supporting clients with a wide variety of analytical services. We have been around for over 35 years now and are based exclusively in the Reading area across 4 different sites. For those of you who don't know where Reading is, we're about 40 minutes west of London and Heathrow. Our 4 sites accommodate over 350 scientists working and supporting clients in 12 different technical areas, of which microbiology is just one of them. We support clients globally with clients all around Europe, as well as the US and as far away as Singapore and Australia. RSL is a trusted CRO with a big emphasis on quality, science and service. This can be shown by our high Net Promoter Score. Because our sites sit close to one another, we can show great agility to manage complex and multidisciplinary projects. We also have a dedicated multi-lab project manager and client liaison managers to ensure that those complex projects are kept on track and managed. So the many technical areas we specialise in allow us to support a diverse range of biological, microbiological, chemical and physical analytical services across all phases of clinical development through to commercial release. We are well positioned to support your project by utilising our state-of-the-art equipment and experienced technical team who can advise and support you throughout your manufacturing. So be that raw routine material testing or investigative support of any issues you may face, such as impurities or foreign bodies in your processes. We have a well-equipped and extensive microscopy department which can help build a specific particle database for your manufacturing process. And our microbiological support is also extensive, where we can support all aspects of your Annex 1 compliance, including microbial identification of any environmental monitoring isolates or unfortunate positives from your bioburden analysis using our state-of-the-art MALDI-TOF system. We can also offer a range of analytical turnaround times to ensure that you get those IDs through as soon as possible to help support your decisions on next steps. So enough of me. I would like now to introduce you to our guest speaker, Dr. Tim Sandle. I'm sure many of you have heard of Tim already and subscribed to many of his publications. So Dr. Tim Sandle has over 30 years of experience in microbiological research and biopharmaceutical processing. Tim is a member of several editorial boards and he's written over 900 book chapters, peer-reviewed papers and technical articles relating to microbiology. Tim works for a pharmaceutical manufacturer in the UK, and he also finds time to be an associate professor at both the University of Manchester and UCL. So now I'll pass you over to Dr. Tim Sandle.
Okay, thank you very much. Hey, excuse me. Welcome to the
Sorry,
webinar.
Tim. You've still got the dashboards and bits.
There we go. Is that okay?
If you move your that's perfect. Lovely. Thank you.
See is it the slides the right way? Yeah.
Yep, all good now, Tim. Thank you.
Okay then. All right. Well, thank you, everybody. Thank you for joining the webinar today. So what we're going to be looking at is contamination control. We'll be looking at a little bit on the contamination control strategy, and then we'll be looking at how we will go about conducting audits of some of the key features of contamination control to help us get ready for inspections and also to stress test our system and to look for gaps and related areas. Obviously, in the time we have for the webinar, we can't run through the entire contamination control system. So I've selected some areas that I think are particularly important. And the act of auditing is very useful because audits can help reveal areas that on a day-to-day basis we might be inadvertently overlooking, and it can help us to just keep strengthening. So one of the other recurring themes is going to be continuous improvement. So if we first of all just consider auditing itself. So ideally we would undertake audits by a competent auditor. So that's someone who's sufficiently trained in the auditing process. Now, if your contamination control group are not necessarily audit trained, then you can pair up an experienced auditor with the contamination control lead. And ideally, you want these people to be independent of day-to-day operations. They've got too much interest, too much vested interest in the process, then there's always a danger of overlooking things. It's very important to be systematic, to see things through and have very defined scopes. We need to document our findings. We need to ensure that our findings are based on evidence. So a good auditor will see something in a document, or they will observe something directly with their own eyes. And every finding needs to undergo a degree of evaluation. How serious is this particular non-compliance that we've identified? And the auditor needs to conclude whether or not the objectives of the audit have actually been met. So there's all general audit things. When we come to contamination control auditing in particular, then our ultimate goal needs to be, are we maintaining an appropriate level of control? Have we addressed everything that is in our contamination control strategy document? And are there any gaps in there that might be relating to Annex 1 itself? So, you know, there's a number of checks and gaps and balances, but if our kind of starting document isn't suitable, then we're gonna miss things. And then the other thing with audits are really important is the planning. You know, what are we going to look at? What kind of questions are we going to ask? Where do we want to unearth information? And we need to be going into the areas. We need to take a sample of each kind of system that we're looking at. So good auditing is also sample based. So we'd ask for one or two things into each area, see if there's any repeat patterns. And then onto the right-hand side of the slide, we have the classic improvement cycle. So we are identifying problems through the audit. We're seeking to correct those problems. We also want to have an understanding of the reasons why those issues occurred. And then we want to be putting our energies in to preventing future problems, because otherwise we'll just end up repeating the same issues over and over again. And also just on auditing, there are different approaches that we can take. So we can adopt vertical or horizontal approaches to audits. So the kind of vertical audit approach is looking at a particular function. So this enables us to keep a close eye over a process. So we might look at the functions, say, I don't know, a microbiology department. Horizontal audits enable us to kind of look at an entire process itself. So how does something get from A to B to C? So we're kind of looking at interconnections, and we're also taking a more historical view. So the example on the slide there might be, if we're going to look at HEPA filter, say HEPA filter testing, we're not just looking at it as what was done last time. We'll be looking at the overall trend. Are we having certain issues of failure? Is our pressure hold across our filter decreasing, which might raise contamination concerns? But here we're talking today about contamination control audits specifically. So a good contamination control strategy will be built on a series of pillars. So we need to ensure that we are risk-based, that we are taking into account that the contamination system should be utilising the principles of ICH Q9, which is the global standard for risk management. Contamination control, and this is something which is also embedded in the philosophy behind Annex 1. What happens in lower grade clean rooms where we get build-up of contamination, then we have the very real risk of that spilling over into higher grade clean rooms. Contamination is not necessarily static in one area. We need to stand back and look at the whole system and try and get as much in the way of early warning and early detection as possible. We also need to be examining where advanced technologies can help us. So this is things like the philosophy that there should be a barrier between people and the process. And we need to ensure that our approach is formal. So we should have a written contamination control strategy or its subparts, and our audit process, our audit function should also be documented, and the strategy regularly reviewed and the audits regularly conducted. We also need to take into account sources of contamination. So unless we have that kind of understanding about, where can contamination exist and where can contamination be easily spread, and what are the vectors for transferring that contamination. So we know that most surveys of clean room activities say that people are the biggest contributors of contamination, particularly skin microorganisms. So how are people gowned? How do people move? How many people are present in a clean room? And where do people interact with product or with product contact parts? So we've kind of got that dimension. We also need to understand water as I've said on these webinars a few times, is that water is not only vital for life and also for microbial life, so it's enabling organisms to grow. It's also a fairly effective vector for spreading contamination through aerosols, through people walking through puddles, turning on taps, leaking equipment, and so on. Air is also a factor because the air is very good at distributing contamination. And where we have surface contamination, we need to be mindful about how could something be moved from one place to another place. So one notorious area for contamination is the wheels of a trolley or a cart, which can spread contamination around quite a lot. So you see here the point I'm making is that we can have contamination existing in one area, but we also need to have a clear understanding about how that contamination can be transmitted to another area and potentially cause product adulteration. So we're going to have different types of contamination. So we have microbial, pyrogenic, particulate, chemical as the main sources that Annex 1's pointing us towards. We're going to have various sources as shown on the spiral diagram, and then things can happen at different time points. So there's going to be points in our process where the product is more vulnerable. It has greater exposure to contamination. So if something's in a vessel and you have to take the lid off that vessel, say to add a buffer, then it's at that point that the risk of the product is at its highest. So a good way to start a contamination control audit is to pre-request documents. So to have a read through and to use this information to understand and get a feel for the facility. And ideally, it's good to have these as pre-requests so that you've read these before you start the audit, because it can vastly help with your audit planning and gives you an idea of either where there might potentially be gaps or particular areas of focus. So I would say read the contamination control strategy for the site. Look at the facility maps and layout. So what type of clean rooms are there? What are their classifications? And then what are the personnel and equipment flows? How many people go into a room? Which way do they go in? How does product come in? And how does waste go out? Then have a look at the differential pressure cascade, because again, that's the control of pressure, the positioning of airlocks are all really important for understanding where the greatest points of contamination risk might be. And then get an understanding of how do people get into the facility through the gowning process, and how does materials get in. And then also consider the facility and process design. Is there a logic to it? So one of the risk factors is having clean equipment close to dirty equipment. So has the facility accounted for that? And is there appropriate segregation in place? And that will also include not only the logic of design, but also in terms of using the most appropriate or best available technologies. So we can't audit everything. Any audit is going to be finite in terms of the time allocated, and the extent of what we can do will depend upon the time of the facility. So to kind of illustrate this, I've selected what I think are the key points. Of course, you might have other points, but this is kind of to illustrate how to focus on certain areas. So what I'm going to consider is how clean rooms are designed and laid out, the levels of environmental control that are necessary to achieve low contamination levels, the type and scope of environmental monitoring, how people are the biggest contributors to clean rooms, sterilisation methods and checks, how aspects of quality control can be best orientated to help improve manufacturing controls, either in process or as finished product tests, something like the sterility test, for example. And kind of also some of the information that QPs need to be aware of when they're considering.
Hi, Tim. Just to let you know, your Teams messages are coming up. Could you just shut down your Teams? That'd be great. Thank you.
Non-sterile products, or we are making sterile products. And the most important thing, and again, this is a theme that I tend to emphasize in these webinars, is that monitoring always comes last. So we monitor in order to prove the effectiveness of our procedural and our design controls. So the biggest emphasis always should be with quality by design, building quality in at the outset, and then having an understanding of why we've picked certain things. So if we take the clean room, the auditor should be able to ask, what is the clean air supply rate? What is the extract rate? And what's the overall number of air changes within the room? Because this is fundamental to the suspension of contamination in the air and the rate at which it is removed. So what's the rationale for it? Why has this room got 10 air changes per hour? Why has another room got 25? What's the basis of that? And then similarly, it's useful to ask about the recovery time or cleanup time. So when a major contamination event happens, there's a high level of particles, how fast can the clean room decay those particles to bring it back to an acceptable level? And then we've also got the pressure differentials as well, which is another important measure. What are these set at? What are the alarm set points? When a door's opened, is there any kind of delay on the alarm? Things like that to help us understand how we're maintaining control. And we also need to make sure that the clean rooms are structurally sound. What are they made from? Are there any areas of damage, like splits in vinyl? A big bad is exposed concrete, and so on. And then, and particularly important for sterile manufacturing, is where are the barriers in place that are, where practicable, separating the operator from the product. So if people are the biggest source of contamination within the clean room, which they clearly are, then how do we control and minimize that through a combination of physical barriers and things like localized unidirectional airflow? And there are some things that, again, I think are quite good as audit specific points. So is there sufficient space in the facility for the type of work that's undertaken? And if there are production increases, so batch sizes, number of batches going through, is that still within the original design space and layout? And then does the overall layout help to prevent contamination? So I mentioned earlier about the segregation of clean and dirty equipment, but it's sort of avoiding that crisscrossing of product and waste, for example. And then what's the level of cleaning and disinfection that is in place? What's the rotational frequency between different disinfectants? And how has that rotational frequency, and the overall frequency of cleaning and disinfection being derived at. We can also ask how humidity and temperature are controlled and what impact this might have on product stability, or operator comfort. Where operator comfort goes out, this is where we can get a greater release of particles from the human body. And then it's also useful to ask about when the last maintenance took place, and particularly major maintenances that might be tied to a shutdown, because these are, again, big areas of risk. Emerging out of a shutdown can bring with it a strong risk of bacterial spores, for example. So understanding what measures were taken, was there a change control, was there a risk assessment, what level of additional cleaning and disinfection was actually undertaken. The audit of the clean room also needs to take into account the classification of the clean room. So is this being done at the appropriate frequency, and what is the data actually telling us? And some of the data is amenable to trending. So airflow velocities, for example, or the pressure across a filter face are things that we can look at to get a longer term view of how well our clean room is functioning. And another kind of good audit question to ask is, what is the frequency of HEPA filter changes? Because a HEPA filter failure is a cataclysmic event, so we need to be changing filters before they fail. But what is that frequency, and what is that based on? And also we'd want to know that we are testing according to the appropriate standard and the most up-to-date standard. So in generally we'll be defaulting to ISO 14644. Do we know that the facility is using that standard, and is it using the current standard, the most up-to-date version? And also looking at clean room reports can be very useful. So a given clean room, how many particle counter locations are there? What is that number based on? How were the specific locations selected? And was risk assessment used to shape that decision? So, often, as I said at the beginning, we're defaulting back to quality risk management. What volumes are taken? Are these sufficient to give an indication of contamination control detection? And for the clean room certification, where we're outsourcing, say, to a clean room contract company, what was the process for approving that company? Who was involved, and do we get a feeling that they are competent? Because we're going to base things like our routine monitoring on the outcome of our classification. And then just on the issue I was talking about maintaining the barrier between people and product, that's particularly important for sterile product manufacturing. And there are important requirements around sterility assurance. So the requirement in Annex 1, for example, is that product and non-product contact parts are sterilised by an independent process of sterilisation. So we can't rely on the conventional decontamination process within the isolator. And when we're looking at isolators, then some good audit questions to come up with are what is the leak rate of the isolator, at what frequency are isolator gloves tested, and what's the acceptance criteria, and what's the failure level. And then where we have automated decontamination in place, such as a vapor phase hydrogen peroxide system, then what is the frequency at which that is done? And again, what is that based on? What is the rationale for that process. And all the time we're considering to what extent are people separated from grade A. So the Annex 1 places a strong philosophical emphasis upon people have no place in grade A. Occasionally people need to access grade A, particularly for the setup activity. So how is that controlled? How well is that scripted? Has that been proven through airflow velocity studies? And looking at how grade A is interacted with can be really revealing about a particular firm's approach to contamination control. And where the auditor only has a certain amount of time, then asking to see the airflow study or smoke study videos can be really useful. So looking at these ideally in the operational state where activities are being performed, and looking at the air distribution can be really revealing. And we also need to consider, are we using appropriate technologies? So there's been big advancements with sterile single-use disposable technologies, and these have huge advantages for reducing contamination. So are we, is the firm adopting as many of those as it possibly can? But although these systems are very good, they do need qualifying in terms of the likelihood of product absorption or with the formation of leachables and extractables. So there are some key questions to ask around that as well. Okay, so we're going to change to a different topic and we're going to look at training. So again, if we roll back with Annex 1, but it's applicable to sterile and non-sterile facilities, there is a considerable emphasis upon personnel training. And this is not only in relation to practical skills, but also to ensuring that staff have sufficient knowledge and the skills necessary, that they have an understanding of basic contamination. So reviewing the annual training programme that is going to give regular updates on sterility assurance, microbiological knowledge, contamination sources, that is actually delivered to the operators doing the job, again, can be very telling, and also gives you opportunity to review audit training folders at the same time. And there may be some other things that you wish to gather in relation to training. So have suitably qualified staff been recruited? Has everyone been trained correctly in clean room disciplines from gowning and behaviours? This issue of basic microbiology, but also extends to basic hygiene as well. So are people washing their hands when they first enter the first changing room, for example? How well are people trained in gowning? For sterile facilities in particular, is there a mechanism for someone to come in and raise a health concern that they may not be able to go into the clean room? And again, for steriles, we've got the necessity for operators to take part in media fills. So taking one of these areas, the gowning qualification, then this needs careful consideration and attention. So we need to understand how people first enter the facility when they go through, say, a clean or classified area, because we're not permitted to present outdoor clothing to grade C and above. We also need to understand the gowning specifications for masks, gloves, suits, and so on. How do we know they are suitable as a means to reduce contamination events? And one of the new requirements, drawing an Annex 1 again, was that operators are supposed to undertake a visual assessment of the integrity of the gown before they enter the actual clean room. So how is that process being done? How is that process being managed? So there's some interesting questions that you can place as an auditor around the gowning process, which will inform again about contamination control. Another area that requires focus again for the sterile, but also it is important for non-sterile. And this is aseptic techniques, because even within the non-sterile facility, how someone is taking a sample will affect the validity and the meaningfulness of that sample. So we should have an understanding of what is the program for those who undertake sampling, those who are required to make connections, those who are required to intervene, say in grade A. So it's going to be useful for the auditor to review at least the C1 activity of sampling, to see an example of how equipment is set up, and if it's a sterile facility, it's always good to focus on the aseptic processing line, and then how people interact with product in containers wherever they need to. How well controlled is that, and are there potential contamination hazards and mechanisms for that contamination to spread? Okay, so moving on to water. Water obviously presents both viable organism, principally bacteria, and endotoxin risks, as in byproducts of gram-negative bacteria. And there are a lot of poorly designed and maintained water systems out there, and there are things that can go wrong, and particularly from our contamination perspective, we're going to have a strong concern with biofilms. So we're going to need to have an understanding of both the way that water is generated and with the sampling outlets as well. So with water system generation, one thing, if we have a water for injection system, then we are permitted to use reverse osmosis or distillation. Distillation is a more robust process. Reverse osmosis is effective, but there are always risks of biofilm buildup on the filter, which then leads to endotoxin risks. Some key kind of audit questions to focus on are with the speed that the water is moving through pipework. This is expressed by the Reynolds number. So the water should be in a high state of turbulence with a Reynolds number above 4,000. And where we have hot water systems, then it's also understanding, what temperature is that water at, and is that sufficiently high to be self-sanitizing? And generally, it's around the sort of 65 to 70 degree mark, that Celsius, that is, that we can achieve that level of control through heat. And we also need to go around checking for dead legs, and we'll look at some examples in a moment, and also how repairs and maintenance are undertaken. But with any audit of a water system, then walking the system is really important. You can't do it as a desktop exercise. So going into a plant room, for example, gives you an immediate feeling for the level of control. It's a good idea to take the facility diagram, a schematic with you, So you can actually then use that to check, is everything exactly where it should be? The diagram has this, but is this in this place? And then look for leaks, because leaks are a problem. One, it gives an idea of the level of engineering maintenance, but particularly leaks in purified and WFI systems. Any leak is a possible bridge for contamination to enter, even if that system is still under pressure. It's not a complete guarantee that you won't get contamination entering the system. It's useful to take into account difficult to take sample points, particularly in the plant area. So if QC come along once a week and they have to clamber over pipes and various things, how can they take that sample in a hygienic way? And also then to check, have there been any recent alarms and what frequency of those alarms reviewed? And then when the actual system is looked at, the user outlets, then you can call upon some critical points and ask to see the data trends for bioburden and for endotoxin, and also keep an eye out for poorly maintained hoses. And on that subject, we have some photographs here. Of some poorly maintained hosing. So we've got hosing that's not disconnected in the top diagram. So when tubing isn't in use, it should be disconnected so it's pre-draining, and the tubing should be changed at regular intervals with freshly sanitized tubing. We've also got again tubing that's not free draining properly. We've also got a picture there of an inappropriate valve that holds water. So that's a huge source of contamination. And then we have an example of a dead leg. So where we have the dead leg, again, we've got a great opportunity for a biofilm to develop. It is also useful to conduct audits of the process of sampling to follow the QC staff around, and then also then to relate that to how production use the water. So if QC are flushing for 1 minute, but the production staff are flushing for a shorter time or not flushing at all, then the QC sample is not representative of the quality of the water that production are using. And then it's also important to understand the sampling plan, how often a sample is taken, what's the frequency, once samples are taken, what's the time limit on them being tested, and under what conditions do they need to be stored at, are the test methods appropriate and validated, and then also to look at how water system excursions are dealt with. What's the procedure? Does an outlet go into quarantine? What is the definition of a problematic trend? There are also issues that we need to consider in relation to equipment. So any pharmaceutical facility will, as you know, contain many items of equipment. Each needs to be well designed, to be validated and controlled. So getting an understanding of, what are the processes in place to control this, and you can pick on individual items of equipment or do a general overview. And it's useful to know that, when does the equipment undergo maintenance, what kind of qualifications are undertaken, how often is it verified. But an important area to ask, and sometimes one which gets overlooked, is where equipment needs to be sent away for repair and for maintenance, how is it returned to the facility, and what's that decontamination process like? This also gives you an opportunity to inquire about the change control process. So if something can't be done that falls under a definition of like for like, how was that change managed? And are there any contamination risks that have been introduced through that process? And some general questions to ask in relation to equipment, as I mentioned, is about like maintenance and schedules and frequency. Also asking about preventative maintenance. So, what level are modifications undertaken to prevent things from breaking down in relation to servicing and calibration, to ask about qualification and verification of the equipment. So if it's something new, you can ask about the installation. If it's in routine use, you can ask to see a number of performance factors around the piece of equipment, particularly where you know there's been an increase in production. So if things have been ramped up in order to get more batches through, what's the impact of that stress on that piece of equipment's ability to function, but also to maintain contamination control? So if we're using a ultrafilter more often, how are we sure that we've got good endotoxin control, particularly if the frequency of sanitization has had to be modified in order to cope with the additional batches that are going through the facility. And we also need to consider sterilisation as well. So where we have sterilisers, the auditor may need to understand what was the selection, design and location of the equipment. You know, what are the cycle programs that are used and how were they qualified and how was worst case defined? And then what are the critical parameters that tell us that the particular sterilizer is working correctly? And then what was the approach to validation? Was this based on product composition? Was it based on mass? Was it based on the dimensions of objects? And what's the logic behind that? So many autoclave validations will work on total mass, but it's been shown through studies that something like a very narrow piece of silicone tubing, which would not weigh very much at all, but targeting heat in the center of that can be severely problematic. And when we've used chemical and biological indicators, then what's our understanding of those? How do we know that they've been used correctly? Did they have the right population, the right D value? And then if you pick on a piece, a sterilizer like an autoclave, then some useful questions to ask are, how has the heat distribution been assessed? What is the steam quality, particularly the steam dryness fraction? How do we know that the air is removed from it? And again, around the qualification method. I also have environmental monitoring. So environmental monitoring, as I mentioned earlier, is not a substitute for poor environmental control. So control always comes first. Monitoring looks for failures in control. So you want to know how a location selected, what is all this based upon, how is data trended, and how is the data used to fit into that idea of continuous improvement that we spoke about. So behind every environmental monitoring program, there needs to be a risk assessment, and that should include things like process flows, justifying locations. It should be based on an understanding of, you know, those hazards that we mentioned earlier. What are the air risks? What are the surface transfer risks? What are the operator risks? And is there a logic to this? And particularly with controls of people and how they move and interact. And another key area in relation to contamination control is cleaning and disinfection. So, you know, what are the agents that are being used? How were they selected? How were they qualified? And do they have sufficient diverse activity that they are suitable to eliminate all of the types of organisms that we might be concerned about? And when and where and why is a sporicidal agent used? Equipment cleaning is an important area, and regulators will focus often on equipment cleaning. And wherever there is manual cleaning, that's always sounds alarm bells. So introducing this into the audit process is essential. So we need to make sure we've got established procedures of cleaning and maintenance, that we have properly qualified cleaning regimes, that we are separating clean and dirty equipment, as I mentioned, and we are avoiding wet equipment at all times. So after we've cleaned equipment, we should have good drying procedures, because wet equipment is a fundamental source of bacterial contamination and also of bacterial endotoxin. And then we have material transfer. And this can be something that's great and always in control, or it can be one of the major sources of introducing contamination into our facility. So how are we getting items in? Are we sterilising items ourselves and using things like double-ended autoclaves, or are we purchasing single-use sterile disposable technologies, which are good in themselves, but how do we get them into the facility? How many layers of wrapping do we have? At what point are we taking those wrappings off? At what point are we using a sporicidal agent? Do we have appropriate transfer hatches with localized air supplies? So mapping that whole process, the auditor following that, again, can reveal rich amounts of information. Okay, and then we're on to my final point that I've selected. And we've got filtration. So filtration, wherever it's occurring, is really important, but we need to have a lot of product knowledge. Is the filter suitable? Will it absorb too much of the product? Will it impart impurities or toxic substances? How has the filter been validated? How are we controlling time and temperature, for example? So do we have all of that in place? And then also asking for data about the frequency of failures as well. Gives an idea of either the suitability of those filters, or at least how those filters are handled inside the facility. So in conclusion, there is no universal approach. Every area is different. The auditor should begin by understanding process flows and risk assessments, to understand how the quality risk management system works, and there should be a real understanding of holistic approaches to contamination control. And my final point is that the audit process should be used to drive improvements. It's not just there to say tick, 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 everything's good. There's always things we can do better. So it shouldn't just be a comfort blanket, but it should be a proactive tool to drive us forward. Okay, so that brings the main part of the webinar to a close, and we can move to the Q&A part.
Thank you very much, Tim, for the information and comprehensive overview. That was super interesting. So I've had a couple of questions come in, so I'll just ask a few in the time remaining. So firstly for Tim, so how can we control the pyrogens other than endotoxins in the clean room?
I mean, pyrogen control requires quite a detailed risk assessment. So the first thing we need to do is to determine whether we have any risk from non-endotoxin pyrogens, or if we're just focusing on endotoxin. And just to, I've not got very much time to answer the question, so if we just focus on, say, endotoxin, that would be about ensuring that the water is not a source, we don't have wet equipment, that our incoming materials, if they're at risk from endotoxin contamination, are suitably tested. And then it's determining where we might want to have endotoxin reduction steps in the process. So we might use ultrafiltration, we might use rinsing, we might use ion exchange chromatography, We might be using depyrogenation if we're making a sterile product with heating vials. So there's a number of factors. It's a very, very broad question. So the key thing is to kind of work on a very detailed risk assessment.
Perfect. Thank you so much, Tim. I'm just watching the time, so I think unfortunately we're very close to time. So any other questions, like I said at the start, we can circulate afterwards. So I'll just wrap up now for everyone. Just go to the slide. So thank you very much for attending our webinar today. I hope that you found it very useful, and we really appreciate the questions that you've asked. And like I said a minute ago, any questions you didn't cover during our Q&A session, I will forward to Tim, and I'll circulate these along with the recording afterwards. And if you wish to catch any of our previous webinars or Tim's blog or regulatory update, please go to our website and keep an eye out on our social media posts. We're always posting on LinkedIn and stuff, so make sure to hop on over there. We also offer a range of technical training courses designed for professionals across the industry. We've got several upcoming sessions as well, so including those detailed on the screen, and we've got more scheduled before the end of the year. So you can find full details for these and registration links on our website. And remember to also check our website for future webinars. So thank you very much, everybody, and have a great rest of your day.